In this week's lab, we are going to learn urinary system. So this is an overview of the urinary system. You have kidneys, one on each side. Right? The right kidney is like lower because there is a big organ, the biggest internal organ in our body, liver right here. Right? So it's pushing the right kidney slightly lower. Then you have a ureter come out of each kidney carrying urine away, right? Then empty that into urinary bladder. Urinary bladder sits really low in the pelvic cavity, and then you have the urethral, then connecting the basically drain the urinary bladder out to the external of the of our body. So this sequence, this order of urine traffic, right, from which organ to which organ, so on its way out, that is important. In addition to the urinary system organs, you need to be aware where they are, right, inside of the abdominal pelvic cavity. So not only the urinary organs, but their relative location in regard to the other organs in the in the abdominal cavity. So you have the descending aorta right here, right? And you also so that's carrying the artery, right? The oxygenated blood. So that's artery carrying basically the blood into the kidney, and you also have the vena cava, the inferior vena cava, basically where the renal vein drains into, right, from the kidney. So those two are also connected with the kidney. In addition, on top of the kidney, you also have the adrenal glands, right, one on each side. And you also have, they're all under the diaphragm here. Right? This is a posterior view. You can see there the kidney locates right next to, right anterior to the, right next to the last pair, the 12th pair of ribs. So if we focus on the kidney first, right? So we do a cross section. You can see right here, a kidney is linked to renal artery, renal vein, as well as ureter, all in this region, right? And that's the concave side of the kidney, and that's called the hilum. So let's look at this side. It's more clear on the diagram side, right? You see the cortex of the kidney, this renal cortex, and medulla is the one that's closer to the center, right? So the medulla, you can also see the medulla made out of the renal pyramids. This is a triangle-shaped structure, so it's called renal pyramids because basically the apex of the pyramid is facing to the hilum, right? And so you can see the arrangement of the renal pyramid, right, with all the apexes facing the hilum region. And between the renal pyramid, you have this structure called, it's essentially like a column. So it's called renal column. Those are the structures that's sitting between the pyramids. Okay. And so again, the kidney, because their function is to filter blood, right, to get rid of the waste and metabolic waste and other toxins, right, and other biological active molecules. So it's highly vascularized. So the next slide we're going to go through how the blood vessels are going to travel. But this particular one, we focus on the kidney structure themselves. So you can probably see at the tip of the renal pyramid. So that would be called renal papilla. And renal papilla, at the end of the renal papilla is where the renal urine is the end generation of the urine, right? And so that's where the first part of the urine formation. And the urine, once they're in the renal papilla, we call it urine, right? So the urine are going to be drained from the renal papilla into the small canal, and that is called minor calyx. And then the minor calyx going to come merge together to become major calyx, and the major calyx then going to continue to converge. Then it becomes the biggest part right before it emptied into the empty into the ureter. It is called renal pelvis. Okay, so that's on the diagram side. If we look at the dissected side, right, you see the renal pyramid here, renal column, right, at the end of the renal, the apex of the renal pyramid, that's the papilla. And the papilla is right next to, drains into the minor calyx, and then minor calyx merge into major calyx, right? Several minor ones drain into one major calyx, and then it goes into the renal pelvis, right? Then the urine is going to flow out into the ureter. Then next, let's look at the anatomy of a nephron. We talk about the nephron is the functional unit of a kidney, right? So with the nephron, right, you start with you have this hand in a glove situation, right? So in the baseball glove situation, on one side you have the glomerulus, right? That's the capillary component of the nephron, and then around that, right, you have the glomerular capsule around the glomerulus, and together they make the renal corpuscle. Renal corpuscle is the first part of a nephron, and then so the filtrate basically going to be generated in this compartment, right, in the glomerular capsule, and then going to flow into the tubules, nephron tubules. And the first part of the nephron tubule are called proximal convoluted tubule, and then so that's PCT, and going into the nephron loop. The nephron loop can be divided into two parts: descending limb first, and then ascending limb second, right? And then it's going to then go into the distal convoluted tubule, then into the collecting tubule, and then collecting ducts. So this sequence, right, the flow of the filtrate, the fluid inside of the nephron, so the sequence of the tubules it goes through is important. You need to be able to put them in order and put all those different parts on the diagram like this. So that's why in the exercise 8, page 2, it asks you to draw a nephron, okay, on a piece of paper, and then label all the listed structures in that page. And so that's part of your homework. You need to do that. I'm going to collect that as the assignment, as part of the assignment for this lab. So after we talk about the flow of the fluid, right, renal fluid in the kidney, next we need to talk about the blood flow. Okay, the blood flow started with renal artery, but you can probably get from this diagram that for the most part, the artery and the veins are arranged, are organized, they flow side by side. So the way, the kind of the order of the blood vessels they go through, therefore they have shared name. Okay, so the first part is you're going to have segmental artery, right? Which you don't have that corresponding in the vein, but starting from the interlobal artery, then you're going to have interlobal artery, and the one accompanying the interlobal artery, then would be the interlobar vein. So we are going to then only focus on the artery for the most part, because the opposite side, right, the one that's right next to it will be the vein. They share a similar name. So we start from renal artery, and then it basically branches into a couple, big segmental arteries, and they're called segmental artery, right? And then so this order you need to memorize, right? The next one up is basically from the segmental artery going to different. It calls interlobar because they travel between the pyramids, pyramids. So that's why it's called interlobar, right? If you consider each one of them is a lobe, then this will be between the lobes, and then it's interlobar. So it's from the segmental artery, goes into the interlobar artery, and from the interlobar artery, you can see it kind of going to travel along this edge. So this is the separation between the artery, between the cortex and the medulla. And so this is, I forgot to mention this so this part, right, that borders the artery, the cortex and the medulla, it is anatomically called the cortical medulla junction. So you have a segment of the artery or the and the veins, right, going to travel in this junction, right, in the cortical medulla junction, and this artery travel this junction is called arcuate vessels. So the red one is the arcuate artery, the blue one is the arcuate vein. So this is basically they travel along these lines, this boundary. And from the arcuate artery, then it's going to branch. So the branch is feed branches, and it's going to actually into the cortex now, right? So if it goes into the cortex, it's called interlobular, not lobar, but lobular artery. So it's between because different lobules, right? So it's called interlobular artery. And from the interlobular artery, you can see the branch here. This is a zoom in the cortex and the cortical medulla juncture, right? So you have the branch out from the arcuate vessel into the interlobular artery, and then it's going to turn into afferent arteriole that's going into the glomerulus. Then you go into the glomerulus and come out still as an artery. Right? This is very unique of this arteriole. So it goes into a capillary, but then comes out still as an artery. Still carries oxygenated blood in this case, and it is efferent arteriole. And then from that, it's going to go to the capillary bed. So you have the peritubular capillaries, and you have the one right next to the nephron loop called vasa recta because it's going straight down, right? So vasa recta, and that's associated only with the nephron tube. Sometimes it can go really deep into the medulla, and they have important function, right? The medulla, the nephron loop as well as vasa recta in setting up the concentration gradients in the medulla. So that's the part we talk about in the lecture. But then from there becomes then a vein. It's going to go back to the heart. So the blood is going to drain into the interlobular artery. Interlobular vein. So it's going to this vein, interlobular vein, and then going backward, right? Going to the arcuate vein, then to the interlobar vein, right? Then going back, then to the renal vein. So this sequence, where do you put the order, right? If I am pointing out on a model, right, a particular blood vessel, you need to be able to tell me whether it's artery, whether it's vein, right? In the kidney, if it's blue, then it's a vein. If it's red, that's an artery. But then not only that, but what kind of artery or vein that is. So you have to know this diagram really well. So the order of not only the order, but to be able to tell on the diagram or on the model. So for the histology, in this lab, you only have 2 slides. Right? The first tissue is the kidney cortex, showing you here in this diagram, in the figure 24.6a. Okay? So 24.6b is showing you the medulla. In the medulla is mainly just the ducts. So different sizes, because some of them is collecting ducts, some of them is showing you the nephron loops. So it's a little bit hard to distinguish. I want you guys to really focus on the renal capsule, because here you can see the ducts, but also you can find the glomerulus, the renal copsicle. Right? You can only find the renal copsicle. That's the part here, copsicle, right? Including which includes the glomerulus inside. You can only find that part in the cortex, not in the medulla. You can see the twisted around capillary vessels. In addition, there's also a layer of the visceral layer of the capsule right here. Then you do see a space. Can you see a space, right? Then between the visceral layer of the capsule, and then you have a parietal layer of the capsule. So that's that should be a squamous, simple squamous type of epithelium. The cells really look like they are really flat with a flat nucleus, right? So that's a renal corpuscle, and you also see the cross section, right, of a bunch of ducts. That should be DCT and PCT, right? You can't really distinguish. Well, I'm not going to ask you to distinguish which one is which, but you should be able to tell, right? It's kind of, how do you tell? It's a kidney tissue, not another type of organ, not another organ, because if you see the glomerulus or the renal copsicle, then you see the cross section of a bunch of ducts. Okay, that is a kidney. In terms of the other tissue, right, we are going to look at is the ureter. So if you recall, ureter is this muscular tube that can basically going to drain, right, the urine from kidney to the urinary bladder. So they actually facilitate the movement of the urine. The ureter facilitates the movement of the urine by sequential contraction of the smooth muscle layer. So that's called peristalsis, right? The same kind of movement the digestive tract used to move the material inside. So therefore it has a pretty prominent muscularis. Muscularis, you can see, right, multiple layers of smooth muscle. It has a mucosa component, right? In the mucosa component, you actually do not see a round lumen, right? Instead, you have the mucosal fold, right? So those are basically the collapsed lumen, right, are squeezed inside. So that's because, right, when you're fixing the tissue, you do not have the urine inside. So that's all. When you collect the tissue, it's all empty. So the lumen collapse. But it also shows only, like if you can imagine when it's full of urine, when it's transport urine from the kidney to the urinary bladder, this mucosa should be stretched out, could be stretched, right? So what makes this particular epithelium is the transitional epithelium, and the same kind of epithelium tissue actually also line up the lumen of the urinary bladder for their function, so then they could expand, right, based on how much urine is going to be stored in the urinary bladder, or is going to pass by the ureter. So if there's no urine or very little urine, they're going to collapse. The epithelium is going to become more, not squamous, but more columnar looking, the top layer. But when it's stretched out, the top layer becomes squamous strip. So that's called transitional epithelium. That you should have learned in MP1. I'm just kind of trying to connect the dot with you here. So this is where you will find this special kind of epithelium, the transitional epithelium, right, in the ureter and urinary bladder. Well, you're going to say, well, there is a tissue looks pretty familiar, like similar to this, would be the esophagus. It's also a muscular tube. It has, it can also have the collapsed lumen, right? How do you distinguish those two? Well, you actually never see the esophagus by itself. It always will be next to a trachea. So if you're giving a real slice, right? If you're retaking this and we're doing the real slide lab exam. If you see on the same slide, right, you see a trachea, then that's esophagus. Otherwise, if it's just by itself, that's ureter. Okay, lastly, some of you have reached out to me saying, oh, we're supposed to do a filtration experiment, and how are we going to do this? Well, I try to find whether in the in vitro lab kind of a session at Connect, whether they already have something made up. It looks like they don't. So I'm going to actually, this is a really simple experiment. You can adopt it, I already kind of set up a at home, like a experiment version, right? Let me walk through what we are supposed to do if we do it, in the lab, right? We're going to set up a funnel. We're going to have a beaker to collect the fluid that filters through. The filter, of course, right, a filter is going to be put in the funnel, right? And so what we are going to filter through the filter paper is we're adding a kind of mixture with three things in it. It has water as a solvent, right? Then it has the copper sulfate. This is kind of blue kind of color, like ions, so it's going to dissolve in the water and give the water this blue kind of color. So it's easy for you to see whether it filters through or not. And then you're going to also, in this picture, have some charcoal. So charcoal, you don't know what charcoal is. So they're kind of you can see is powder like, even though it's fine powder, but you can really see them with naked eyes. So they're not dissolvable in water. They stay as a charcoal because, charcoal are non-polar, so they're not going to dissolve. They're big, right? So you can see them by naked eye. So they're going to just kind of, it's a suspension. If you mix it, swirl the bottle, right? It's going to, be suspended in the water solution. So that's what you put in the funnel, right? So you're going to put it in, right? Forgot the volume, maybe 20 or 30 ml of the solution. And then you're going to wait, and you're going to observe the substances, right, going through the filter paper, and you're going to look at what's in the beaker, right, because what's in the beaker, that indicates that it got filtered through, and so you can kind of tell from this diagram. Did you see the filtrate? So filtrate is what after going through the filter paper is called filtrate, right? So do you think the filtrate has blue color or has copper sulfate? Looks like yes, right? Strong in blue. And how about charcoal? Can you see the black dots charcoal powder in the filtrate? The answer is no. So we're supposed to do this, right? Then you're going to say, okay, what filters through, okay? And copper sulfate. Why did it filter through? Because it's a small inorganic molecule, right? And so what did not filter through? Charcoal. Why didn't it filter through? Because it's big, right? The filter is a selective permeable membrane, right? It essentially functions as a selective permeable membrane. So it's going to stop some things from going through it and let other things go through it. And in terms of our filter paper, it's based on the size of the particle. So if it's small, it's going to pass. If it's too big, right, for the hole in the filter paper, then it's going to not pass. So that's why, we don't see charcoal in the filtrate. But then the other question is then we're setting up, not only for filtration, we're kind of setting up this for kind of modeling what happens in the nephron. It's specifically in the cortical neuron, in the renal cortical. You remember in the renal cortical, right? You have the glomerulus carrying the blood in, and the filtration process happens, right, in the cortical. So what filters through? The blood, right, filter through the filter membrane. In the lecture, we talked about what are the cells that made up, the two cell layers, right? One layer is on the capillary vessel wall, and the other is the podocyte, right? Those spread out cells that sit on the basement membrane separating the blood vessel, right? The capillary vessel from the podocyte. So this is together, the three components together make up the filtration membrane. That's equivalent to our filter paper here. So what we put in is essentially what we have in the blood. So then basically these models, right? The funnel models the and plus the filter paper models the capsule, right? You have the visceral layer that's semi-permeable. You have the parietal layer, that's the funnel, the glass funnel, that's completely impermeable, right? So the result is then the blood is going to pass through the inner one, the filter paper, and then generate a filtrate, right? And some of the things are going to be left behind, such as big molecules, formed elements, right? The big proteins, and the small molecules going to pass through. It's just like what we have seen here. So that is kind of what this is modeled for. Okay, at home, can we do this? Yes. We can. You all have, brewed coffee, right? Before, either at home or at least if you don't have a coffee maker at home, you probably have seen other people making coffee. If you don't have a coffee maker using, a filter paper and coffee powder to brew coffee at home, then you can probably search up online videos and find some videos people brewing coffee. I'm sure there are other videos out there. The setup is like this, right? There's one more question I want you guys to, one more thing we didn't mention other than what these models and what's filter through, what does it, why does it filter through is the filtering speed, the rate of the filtration happens. So you're probably, if you can do this at home, you can say if you basically I'm not asking you to make coffee, you're doing experiment, right? So you should try to put, instead of letting the coffee maker itself slowly put in boiled water to the coffee powder, you should try actually to dump, a cup of water or half a cup of water into the filter paper with the coffee powder inside. It will be essentially the same as here, right? I want you to observe how fast, right? Not too much. You don't want to dump too much, so then the overflow happens. But then I want you to dump a reasonable volume, and then you can observe how fast it happens. Is the filtration rate the same in the beginning or is in the end? If there's a change, right? It become faster or it become slower? And what do you think that's basically drives the rate change? So that's basically similar like what we have in the nephron. It's a filtration rate. So what's driving the filtration rate, right? In our nephron, and you can try to model this. So I want you guys to answer the following questions. That's in the next slide. Right, so there are 5 questions. The 4 and 5 has 3 parts, but we kind of went through that already. But the first is the cell structure that the filter paper represents. Right, we just talk about. Right, the filter paper represents what, and then the force that drives the filtration rate. Right, so if you observe the changes, right, in the filtration rate, then why does it change that way, right? You can talk about the filtration rate is in your at-home experiment. What do you think drives how fast the water, basically water going to pass the coffee filter? And then the 3 parts question you try to answer, right? We kind of went through that already, but I want you to give examples of the substances in the blood behave like either, the thing that not pass filter through in your experiment, or what is, filtered through, right? In your experiment would be coffee, right? Then give some example, then what's in the blood can be filtered through. Okay, so I want you to submit this as well as your drawing of the nephron and the histology drawing, all of that. So that's your basic experiment lab assignment. Submit that through Google Classroom, and I'm going to put up exercise 8 classwork, right? So then that's where you'll submit that to. If you have any question, you can ask me, in our virtual time.